Okay, hello everyone. I'm going to be teaching the biology course today, so let's get straight to it. Today our topic is classical genetics. Um, so basically I'm going to be going over some of the aspects, simple classical genetics, crosses, and things like that. So hopefully it'll be useful to you guys in the future, whether it be for AP Bio or like your pre-AP, honors biology, whatever. Okay, so as a brief overview of what I'm going to go over today, we're going to be first going over why genetics is important. And then we'll be going over the beginnings of genetics in Mendelian principles, as well as some of the modifications to Mendelian principles, as well as extensions and linkage. So first, let's go over why genetics is even important and why scientists want to study it. So genetics, the usefulness of genetics is hard to overstate. It's could be considered maybe one of the most important fields of biology because its applications are so widespread. For example, genetics is used whenever people are doing family planning and they're looking to check if their unborn baby or potentially if they want to have a child would have a disorder of any kind. Um, it has potential to bring cures for many diseases and many causes of diseases and syndromes and disorders. They all lie, their root is in some genetic error. As well, genetic engineering is highly important, as you know, for GMO crops, which although GMO crops are very controversial right now, it's impossible to deny that they have a significant impact on the food security of the United States, as well as other forms of genetic engineering. Because even domesticating crops, like, for example, corn from teosinte, because crops that you eat nowadays, they aren't just found in the wild, they had to be domesticated first. That constitutes a form of genetic engineering, albeit a very slow one. Okay, so and maybe the most important thing is that genetics is important to reduce human suffering. Most drugs that exist nowadays, as well as cures for diseases and different forms of beneficial things to humans, they all come from genetics. One example is sickle cell disease, which has a genetic basis. It's actually only one codon or one nucleotide in DNA is changed. So it's like one base pair, which causes such a debilitating disease to so many people across the entire globe. So this is why we must study genetics. Okay, so first we're going to be going over Mendel's experiments. So Gregor Mendel was um, a priest in a church in Austria, in Vienna, I believe, and he was very well known as the father of genetics. He's the father of genetics to all geneticists nowadays. And he began the study of genetics in the back of his church by growing pea plants. So basically what he did was he grew many plants in order to figure out how traits among the plants were transmitted from parent to child, okay? And he did this with several different traits. In pea plants, he did several with flower color, like plant height, um, like pea pod shape, pea pod size, pea color, and pea pod color, and things like this. And all the traits he tested had had several major characteristics in common. One is that they were clearly found in two different traits. For example, for flower color, he found flowers that were either purple or white. So this flower color trait didn't have any sort of gradation in between. It was just either they were a white flower, either they were a purple flower. So this applied for other traits such as plant height. He found tall and dwarf plants, or pea color, found green and yellow peas. And he named that these different traits, different things, which we still use nowadays. He named the trait that was found in the F1 generation, which I'll explain in a bit, dominant, and the trait that was not recessive. So basically what Mendel did was he took some true breeding lines of peas, and true breeding means that over subsequent generations, he would breed these plants with themselves over and over again, and they would continually retain the same traits. So for example, the purple flower in the top of the below parental generation, that would be a true breeding line. He would continually breed these and they would only give him back purple flowers, okay? So that's how you know it was a true breeding line. He did the same with the white flowers, right? And what he tested is what would happen if he crossed these two true breeding lines. And the parental generation, or the P generation, is composed of these true breeding lines. And then he called the second generation, or the generation that came from these parents, the F1 generation, or the first generation. The F stands for filial, which in Latin is like means son or offspring. So he called this F1 generation the first generation, and he found that in all of the traits he tested, the F1 generation only had one kind of trait. So, for example, in this example, the F1 generation was all purple flowers. It wasn't somewhere in between purple and white. It wasn't entirely white either. All the flowers were just purple. And this was contradictory to what was believed at the time for Mendel's time. So at the time, people didn't know anything about genetics, and they believed in a form of inheritance called pangenesis, as well as blending inheritance. So basically what people thought was that if you mated two animals or plants together, the offspring would look like a blend or a mix between the two parents, which isn't entirely false, but it does not give the full picture, and it's not true for most genes. And we'll see later, especially tomorrow, that at a molecular level, it's not true. But for example, if you were to look at people like your friends, your family members, you would probably come to a similar conclusion, right? Like you probably look very similar to, like, if your two parents were combined together, like somewhere in between your two parents, and your friends probably look the same, right? So a person might come to this conclusion, but Mendel thought that this conclusion was incorrect, and this experiment clearly demonstrated it, because the F1 generation had the same color as one of the parents. It wasn't in between, okay? Now, after he crossed the F1 generation, or after he crossed the parental generation, he decided to further mate or cross together the F1 generation plants to see if there was any more to be learned. And what he found was that in the F2 generation, some of the plants had white flowers. Most had purple flowers, but some had white flowers. And to Mendel, this was another important result of his experiments, which disproved a second part of his the commonly found beliefs at the time. Another commonly found belief at the time was that people believed that since there was a blending inheritance, they thought that, for example, if your mother doesn't have a particular trait and your father doesn't have a particular trait, it's impossible for the child to have a trait. For example, they believe if the mother doesn't have blue eyes and the father doesn't have blue eyes, it's impossible for the child to have blue eyes. And Mendel's experiment proved that this wasn't the case, and that genes were more complicated than originally thought. So Mendel, without any knowledge at the time about any of the molecular basis of any of his experiments, he guessed actually, or he managed to propose a theory about how these genetics were inherited, and he actually proposed a correct theory, which we still use now today. And his theory was that each organism has two alleles of a particular trait, and alleles basically means that he has two variations of a particular trait. So each animal or plant, for example, in this example of plant, will have two alleles, which can be dominant or recessive. And the combination of the alleles determines what the organism's outside like appearance, or their phenotype in Mendel's terms, is. So in this example, there are only two alleles, which can be the uppercase or lowercase b. And scientists use two terms to differentiate between the different kinds of combinations of alleles that a person or an organism may have. They call it, whenever the organism has two of the same kind of allele, they call the organism homozygous for this allele. And if they have two different kinds, they call it heterozygous. So in this example, the parental generation is homozygous, homozygous for their respective alleles because they are true breeding, while the F1 generation is all heterozygous. So with this in mind, we must now figure out or talk about what is the genetic basis, the molecular basis of the things that Mendel discovered. And for this, we'll have to keep looking further into the laws of segregation and independent assortment. So basically, the law of segregation, which Mendel discovered, states that whenever a cross happens, so the parent which has two alleles, into their gametes, the alleles segregate. So as you can see in the picture below, or the picture to the right, there's a segregation between two different kinds of alleles. So you can think of it as the red and blue alleles, right? They segregate into different gametes or sperm and egg. And as a result, the two alleles are not going to be transmitted at the same time to the offspring. So as a result, this means that the separate results that he discovered were as a result of the law of segregation. Okay. Mendel also conducted experiments on other kinds of crosses. Originally, in these crosses that I illustrated below, over here, he only crossed one trait at a time. However, he also tried crossing multiple traits at a time to discover how these traits were related. And he found that there's a law of independent assortment. And what this basically means is that whenever two different genes or traits are crossed, the different alleles for each gene segregate independently, and that is that they will distribute independently into gametes, law of segregation, without regard for the other gamete or other allele. So as a result, he found that a 9:3 to 3:1 ratio resulted. So in this original monohybrid or one trait cross, he only found there was only a 3:1 ratio, but in the dihybrid cross or the two trait cross, he found that there were a 9:3 to 3:1 ratio. Okay, moving on, we're going to discuss a bit about some modifications to Mendelian inheritance. So basically, there are some difference in Mendelian inheritance that subsequent scientists found. So they found that Mendel's experiments weren't all encompassing and they weren't completely correct. So some of the modifications that they found were that different kinds of traits can be inherited in different ways. For example, incomplete dominance. So in some traits, for example, in flower color in certain kinds of flowers, it does seem to have a blending mode of inheritance, which means that if there is a red true breeding and a white true breeding flower, their offspring will all be pink, which appears to support the blending mode of inheritance, but actually this mode of inheritance in complete dominance is not blending at all. And basically how it works is that the heterozygous organism has a different phenotype than the dominant or the recessive homozygous. And as a result, it's not really accurate to call it the blending mode of inheritance. There's also codominance, which is a different mode of inheritance where the offspring in the F1 generation appear to have both of the parents' traits. So in this example of codominance, the red and white flowers produces a red and white flower, or the flower has both of the parents' traits. Okay. So finally, there's also an issue of penetrance, which is basically because genetics is extremely complicated and there are a lot of different gene interactions, some genes aren't fully expressed. Even if they're dominant or recessive, a dominant gene may only sometimes be expressed. And this is called penetrance. Okay, now let's discuss a little bit about linkage, because linkage is an important aspect that seems to contradict what Mendel originally stated, but is actually merely an extension. So for one, it's important to realize that genes are located on chromosomes, okay? So since genes are located on chromosomes, and there tends to be many more genes than there are chromosomes, so multiple genes must be located on a chromosome. As a result, because of the molecular um, basis of inheritance, which we will talk about more tomorrow, um, the genes may or may not segregate independently, as stated in Mendel's law of independent assortment. Okay, so as a result, some genes may be linked together, and what this means is that certain alleles tend to be inherited together in patterns, and this is due to a combination of meiosis as well as um, the genes are located close to each other on the chromosome. So we'll be talking about this more tomorrow, but this is just a brief overview. Okay, so now we are going to play a short Kahoot. So um, let me open this Kahoot and then we get right. All right, so y'all just join this code. Okay, is everyone joined that wants to play in the Kahoot? If you're not, please text in the chat, or else I will be starting it very soon. All right, it looks like everyone. Oh, okay, one more. Okay, I guess that's it. If you guys, I'll put the code in the chat. So if you want to be able to join later, there it is, and we'll start. Okay, it looks like mostly everyone got this right, and the answer is dominant. Okay, so maybe some of you did not understand the meaning, and that's okay. The question is asking basically what um, form of allele can only be seen whenever there's a homozygous, it's recessive, because a dominant trait can actually be seen when it is heterozygous. Okay, so I'm gonna talk about this one a bit because most of y'all got it wrong. So I didn't discuss this a lot in this PowerPoint because I'll discuss a little bit more tomorrow, but in fruit flies, they have the same form of inheritance, sex inheritance as humans, which is that males are XY and females are XX. Uh, Barring like rare genetic mutations, the male fruit fly passes on the Y chromosome to his son because they only pass on one of two because they are considered pseudo homologous chromosomes. So as a result, the male fruit fly will get one of two X chromosomes from its mother and then a Y chromosome from its father if it's male. So the answer is no. Okay, I'm gonna talk about this one a little bit as well because most of y'all got it wrong. So Mendel's initial experiments had to do with complete dominance. All these modifications to his experiments, like incomplete and codominance, they were discovered by later scientists, not him. Okay, it looks about half of you got this wrong too. So one important thing to note, which I'll be going over tomorrow as well, is that from a inheritance standpoint, it depends what level that you're looking at. Um, in order to determine what form of inheritance it is. But for the sake of this, or this form of inheritance, it is considered incomplete dominance because the pink is between red and white phenotypes, and it doesn't contain both parents. But at a lower level, it may be like at a molecular level, it may be considered codominance, but at a organismal level, it's incomplete. Okay, since most of you guys got this wrong, I'll be trying to explain it. Okay, so basically RNA is a main component of like some structures such as ribosomes and is formed from the DNA, but the actual chromosome does not contain any. It is made of a complex of DNA and protein. Okay, congratulations to everyone. Um, yeah, that's about it. And for this particular Kahoot, it was mostly vocabulary rather than actual problems. I'll be going over some actual problems so that we can do or practice what an actual genetics problem, classical genetics problem is like. So I'll be trying to make some polls and see if y'all are able to do it. Okay. Okay, so I want y'all all to respond to this. Privately message me with your own answer and see how many of y'all. Yes, someone raised their hand. Okay, I guess it was an accident. Okay, I want y'all to all privately message me, answer this question. Um, it's a simple genetics question. Let's see how you do. So the question is, I'll type it out in chat as well. The question is that you have a flower, um, which contains 3 petals, um, and is from a lineage of flowers that is true breeding, and then you want to breed it against your uh flower that has 5 petals, which is also true breeding. Assuming that this is complete dominance and there's only 1 gene and 2 alleles, um, do the genetic cross, assuming complete dominance, and tell me what is the um, result that is likely to happen in the F2 generation. If possible, I'd like you guys to privately message me. I've only got 2 answers so far. I want you to tell me what is the result. What does the F2 generation look like? Good question. The dominant trait is the 5-petaled flower. I don't know if you could hear that, but the 5-petaled flower is the dominant trait. I see a lot of answers coming in, and surprisingly, or maybe strangely enough, or I don't know if I worded the question correctly, the result of the F2 generation. Um, so everyone check their answers and make sure. Okay, I don't know if y'all are all done with your answers, but the answer is that, at least seeing in chat, actually, surprisingly, none of you guys are correct. Um, the F2 generation will have both traits. And going back to the slide, the F2 generation will have both traits in a ratio of 3 to 1, assuming complete dominance and assuming, so there will be flowers with 5 petals and flowers with 3 petals. And there will be 3 flowers with 5 petals on average for every one flower with 3 petals. That was the correct answer. Okay, next problem will be a little bit easier. Do this cross and tell me the results. Okay, I don't know if y'all all privately got your answers, but to Winsu, you're right. The ratio would be 2 to 2, or you can simplify it down to 1 to 1 ratio of AA with upper like a heterozygous to homozygous recessive. Now we're going to do a little bit more complicated crosses. And just give me the phenotype. Please tell me if y'all like need more time, by the way, because I'm not really getting a lot of input from you guys, so I can't tell whether or not you're running out of time or you have finished the problem. Okay, just some hints for this problem if y'all are having a hard time. If any of y'all have ever done competition math, like AMC or anything like that, you can treat it as a probability. You can separate it into two crosses. The A crosses, so heterozygous A cross with heterozygous A, and heterozygous B cross with heterozygous B. And you can make a tree, um, because each of them you can treat individually. That's a hint for you guys if you are having trouble. Okay, if you need more time, let me know, or I'm going to explain how to do this problem in about 30 seconds. So, okay, if y'all are all done, then I'll explain the answer, and the answer is that there will be a 9 to 3 to 3 to 1 ratio of AA, or I'll just type it so I'm not just that is the ratio. And the reasoning for this is the dihybrid cross that I talked about in the earlier slides in the law of independent assortment. And the ratio is 9 to 3 to 3 to 1. I don't know if you guys remember me mentioning that, but that is the answer. Okay, so for the sake of classical genetics, the problems will be similar to these. If you have more questions, feel free to email me. But it's really hard to explain any semblance of like useful information in such a short time period. But for today, I just hope you remember some basic tenets of genetics and why it is important in case you're interested and want to study it further. So actually that will be it for today. We will be ending a bit early, but that's it for today. So thank you all for listening and participating, and that's all. Thank you.